Hello everyone, I'm Juan and I'm also part of the Slovenian lab, now doing my PhD under the supervision of Meta. And today I'm going to talk a little bit more about vibroscapes, in this case active space of substrate-borne signals in a hay-meadow habitat. Now I have to say I'm very jealous of all of you that have been doing experiments in the lab. It's been like 3 years, I did it the last one, and I miss the cleanness, I miss the simplicity of it. Now I'm dealing with this every summer and it's pure chaos, but I'm not here to complain. I need more than 7 minutes for that. So let's talk a bit about active space. It's a concept that is very well studied in the soundscape, and it's basically the distance from an emitter, from the source of a signal, where the intensity of the signal, the amplitude of the signal is loud enough to trigger a behavioral response in the receiver. In more simple words, it will be all of you are right now within my active space because you can hear me, people outside are not. This is, the active space depends on the amplitude of the signal when it's emitted, of the attenuation of the signal through the medium, of the level or the amplitude of the background noise, and also of the sensitivity of the receptors, of the receivers. But in the vibroscape, there is not such clear relation. The active space changes and is highly variable because we have to have in mind other many factors. It is well accepted that in grasslands, the active space of vibrational signals is contained within the plant where the insect is signaling, but we can have also plants touching each other, so these signals can be perceived in nearby plants. We have also touching underground from the roots, and the signals could transmit also like that. Even small air gaps between plants are enough to transmit these signals, and then we have the different plants that we can have in the different morphologies of all these plants. So it's not simple, that's for sure. This is where I was working in the meadow that Rock mentioned before. And as you can see, it's a very heterogeneous habitat where everything is touching everything and very grown. And here is where we had to work, and this is our idea for how to measure the active space. So what we did, we used all of our laser vibrometers from the lab. I was dealing with 5 laser vibrometers, and one of them was pointed at the center of an arena. That laser never moved. It was always pointing at the center, and then the other 4 lasers were pointing at the corners of a square of increase, larger area, right, farther away from the middle point. This is how it looks, more or less, in the field. This is another picture of the setup. So our experimental design, we did 4 experiments. We were measuring between 11 and 1 during 3 consecutive days. And for this, I analyzed 1 hour of every of these experiments, and as you can see, we have 4. The shortest distance is every point, it's away from the center by 5 centimeters, 10, 20, and 30, so larger and larger areas. This is more or less how it would look like. And let's go with results. I'm going to move myself a little bit. So this is at the shortest distance. We can see that here in percentages, you can see from the total amount of signals that I managed to annotate, 190 in this hour. Most of the activity came from the left side of this area. So 62% of the signals were picked by that channel, right? And we can see that I found here 9 different VSTs, so vibrational signal types. And more or less every channel has the same amount of VSTs recorded. As we go larger, we can see already there is a shift. The right side is more active now, and we can start seeing more variability in the numbers of VSTs. We see that some channels start presenting some unique VSTs. Those are signals that were not perceived by any other channel or experiment. And already we can see that the number of different VSTs has increased in this experiment. When we go to 20, we also see that the variability is still there. There are more unique signals. We are recording more things. And on 30, the largest distance, we can see now that the amount of signals, different VSTs recorded, is the highest of all of the experiments. this is a general picture of how the thing looked like. Now, speaking about the vibrational signal types that we recorded, we can see that as we increase the area of recording, well, it is clear that we are going to encounter bigger amount of different VSTs. A nice thing to see is that the total number of VST was 26, but we could see that 2 of them appear in every channel and in every experiment, and these are these 2 insects. We don't know which one this is, so I just name it Rattle. And this one, we are pretty familiar with him. So these insects appear in all of the experiments. So the factor that I would like, the parameter that I use to kind of have an idea of how this active space would look like is the presence. Now, what is presence? I have presence from 1 to 5. Present ones means that the signal that was recorded appear in only 1 channel. Presence 2, 2 channels. Present 3, 3 channels, right? We can see that in the 2 first experiments, the closest distances, while signals that appear in only 1 channel, in the case of 5 centimeters, was 22%, the rest appear in 2 or more channels. 78% of the signals appear in 2 or more channels, meaning that those signals were within the active space. I mean, all of our recording points were within the active space of those signals. At 5 and 10 centimeters, we can see that we pick many signals in many channels, but the drop from the experiments at 20 and 30 centimeters is huge. Almost 100% of the signals were picked only in 1 channel. We can see that there are very little cases where they were picking 2, and who were these? Well, these 2 signals, actually. So as a conclusion, this is bad expressed, the first one, but the amount of VSTs and different VSTs that we encounter increases with a larger area of recording. The presence drops to 1 channel when the points of recordings are situated at distances of 20 centimeters or more. It's highly variable. In every of the experiments, you can see that there is a lot of variability and complexity, and there is more to be done, and there is a lot of data, and I'm on it. But as you know, it's very tedious, especially working with 5 different lasers. But hopefully, AI will solve this at some point. I need it. And the next step is we already made the recordings with our calibrated equipment, a sound book that will tell us exactly what's the drop of amplitude of every of these signals. That's all for me. I would like to thank you for your attention. I would like to thank my lab for the amazing support. This is my Twitter, and before I have the VibroLip, this is an account where I'm updating all these VSTs I encounter in the field, if any of you are interested to listen to a little bit of this. And for the experiment that we saw here today, these are the available VSTs that I recorded if anyone is interested in listening to that. Thank you.